All right. All right, guys. So we're going to go ahead and wrap up our metabolism content for today. And then at the very end, there's going to be a survey that I'm going to have you guys take that's actually tied to the entire BME curriculum. We were going to have Shannon Barker come in today to help administer the survey, but she's out sick today. So I'll help you guys with that. But largely, it's something that we have second, third, and fourth year students do at the end of each year, just to try and get some feedback on general BME curriculum and how things are going, for lack of a better phrase. So we last left off kind of piecing everything together from glycolysis, the TCA cycle, and oxidative phosphorylation. And we'll be bringing that all together, continuing to leverage. I'm going to take a step back. Continuing to work off of this image to the left here to try and understand how different different diets or different macronutrient balances can drive and alter different metabolic pathways. And so for a long time, an old standard diet-wise was that that people needed to consume a low-fat diet. That's what a lot of, I would say primarily from 1980 to the year maybe 2000, that was kind of what the standard prescribed diet typically was. And so what ends up happening is that the idea was that with less fat consumption, there would be less fat synthesis that would be stored in the body. But as we appreciated last class, is that there's more than one way where fat synthesis can occur. And so what ends up happening is that when there is an excess of glucose that's consumed, after that glucose has been, whatever glucose is needed energetically that's been used up, and whatever glucose has been stored as glycogen, whatever is left over, is able to be taken from glucose down to produce glycerol, that backbone for triglycerides, as well as going through glycolysis to produce acetyl-CoA that can then undergo the reverse process of beta oxidation to produce fatty acids that form triglycerides that then produces fat that ends up being stored in our fat tissue. And so whenever there's typically, whether it comes from an ingestion, consuming excess fat or carbohydrates, they're still able to be fat synthesis. And so that's kind of what led to the retirement of this idea of a low-fat diet being the healthiest diet to have. We talked about this a little bit, but there's also a starvation diet where a person that's low on carbohydrates, fat, and protein will end up having none of those biomolecules or macronutrients being available. And so what ends up happening is that they'll end up using muscle tissue that undergoes muscle wasting to break it down, catabolism, to produce amino acids that are then used to feed into the TCA cycle and produce oxaloacetate. That oxaloacetate is also able, during gluconeogenesis, to be used to produce glucose. When we had our first lecture on metabolism, where we looked at glycolysis versus gluconeogenesis, when we're coming back up those pathways, some of the intermediates at the early stages of gluconeogenesis included oxaloacetate. So we can produce oxaloacetate from amino acids, and that can either feed into the TCA cycle or be used for gluconeogenesis to produce glucose. And so in addition to that, we can also have fat that's broken down to produce acetyl-CoA, and that'll also be used to feed into the TCA cycle. What's also noteworthy is that that acetyl-CoA can also be used to produce ketone bodies or ketones. And so these are these are energetic substrates that can be used primarily in the brain and in skeletal muscle, these like highly energetic tissues that can be important when there is a shortage of oxaloacetate. And so when there's only so much muscle tissue available for wasting, there's also going to be the use of fat tissue that's going to be broken down to produce these fatty acids that produces acetyl-CoA that can then be used to produce ketones that are then, again, used as an energetic substrate, primarily in the brain and in skeletal muscle. What's probably more, oh, yes, go ahead. Can you say that one more time? Sorry, I couldn't hear very well. Yeah, all right. You're good. Uh, a uh, the next diet that's worth exploring is a high protein, low carb diet. Back in the 90s, early 2000s, it was described as the Atkins diet. Now there's a few different iterations of it that you may be more familiar with. Paleo diet, keto diet, or in a more extreme case, carnivore diet. And these are all kind of different versions or different balances of how much carbs or how much protein, or sorry, not how much protein, but perhaps how much carbohydrates are consumed, as well as whether it's high fat or low fat. But primarily, these are all really high protein diets, um, and what ends up happening is that rather than having an excess of carbohydrates that are used for glucose to feed into glycolysis, we end up having primarily amino acids that are broken down from the diet to feed into gluconeogenesis via oxaloacetate or into the TCA cycle as well. In the instances where these diets have a higher fat content, say, for example, the carnivore diet or the keto diet, those will also use fats to try and put your body in a state of what's referred to as ketosis, where there's such a low amount of carbohydrates available, where your body has passed a certain threshold now, where it's primarily burning fat as a source of acetyl-CoA, or trying to metabolize fats via beta oxidation to use them as a substrate for energy. And so there's different schools of thought as to what that threshold is. By and large, a lot of people will probably agree that typically under 50 grams of carbs consumed per day will typically put one in a state of ketosis where they're usually producing, using fat as primarily the source of energy here, as opposed to glucose from carbohydrates, and developing both acetyl-CoA as well as in some cases ketones from that acetyl-CoA. What is typically historically been the case is that the keto diet was historically first generated as a diet for those with MS as well as epilepsy. Again, because the idea is that ketones are an important energetic substrate used in the brain. The idea is that particularly with epilepsy, having an increased level of ketones would actually help suppress seizures. And so it was originally developed for that, but now is primarily used as a bit of a fad diet to try and, when combined with exercise, to try and lead to weight loss. What ends up happening is that typically individuals who go on one of these high protein diets for a period of time will actually, when they go back to a normal, a quote unquote normal diet or their previous diet, it actually leads to this case of weight cycling where there's the weight will go up and down. And weight cycling can produce some alterations to insulin responsiveness or insulin sensitivity. And so it's an important thing to be mindful of when trying to figure out what's the right diet and how our metabolism can be altered based on changes in our diet as well. Um, Sometimes the best diet to consider is a combination of lifestyle changes and a balanced diet that has an appropriate caloric intake based on activity level and some context based on as well as size and what you do on a day-to-day basis. But again, there's typically kind of a fine balance to strike in terms of lipid to protein or fat to protein to carbohydrate consumption. When combined with all these things in an appropriate degree, it can help lead to lipolysis, which is the breakdown of fat. And so again, the idea is that when there's an excess of body fat, that it can alter your overall body's metabolism to appropriately respond to glucose, appropriately respond to insulin. And again, there's a lot of correlations of different diseases tied to excess body fat, particularly the visceral body fat. If we go back to the first lecture on metabolism, the body fat that wraps around our internal organs. Those are usually most highly associated with cardiovascular disease and cancer as well. And another additional factor to consider is alcohol consumption. Part of the reason why is because alcohol consumption can also contribute to fat synthesis, as well as some of the metabolic byproducts can also lead to, when there's an excess of alcohol consumption over time, can actually lead to disease. And we'll talk about that in a second. And so as an exercise, we had an individual who would regularly consume vitamin water, which is about a couple hundred calories, but is also fairly high in sugar content. And so over time, had this individual cut off vitamin water, reduce that excess sugar, reducing that excess glucose that they were consuming, and over time able to actually see that when removing that excess sugar, that can actually be contributing to when producing that acetyl-CoA can lead to fat synthesis if there's an excess of glucose that's not being burned and already being stored as glycogen. So when you remove that, you can actually see a drop in fat storage, a drop in weight here. And it's not going to be perfect, but in their honesty as well, they said that they went to a couple weddings where there was an excess of alcohol consumption as well. But you can actually see and appreciate how these things kind of tie together, not just at a cellular level, but to a whole organism level as well. So why these instances of alcohol consumption can actually be tied to fat synthesis? What ends up happening is that we talked about the process of ethanol fermentation in prokaryotes. That's starting on the right side and moving left, where in an anaerobic state, prokaryotes will go from acetyl-CoA that's produced that takes acetyl-CoA and produces ethanol. And so typically for our ethanol metabolism, when we're breaking it down and processing it, we're moving from left to right. What ends up happening is that ethanol will be metabolized via alcohol dehydrogenase that produces a reducing equivalent, and that produces the intermediate metabolite acetaldehyde that then is metabolized by acetaldehyde dehydrogenase. Another oxidation event produces acetyl-CoA. And again, that acetyl-CoA can then, if not used as a metabolic intermediate to feed into the TCA cycle or to feed into, to feed into the TCA cycle to produce the reducing equivalents for oxidative phosphorylation, that acetyl-CoA can be used to produce fatty acids that is stored as triglycerides and leads to fat accumulation. And so, already just mentioned all this. But again, these actually are producing, when we're going to acetyl-CoA, produces the reducing equivalents. And the key thing to note is that acetyl-CoA is the raw material for fat synthesis. In addition to fat synthesis, the more well appreciated side effect of excessive alcohol consumption is that it makes you sick. And that's largely because of this intermediate acetaldehyde that is not a normal byproduct in mammalian biology. And so that's the toxic element that'll actually lead to an individual getting sick and the much appreciated hangover as a response to excessive alcohol consumption. And so what's important to note is that people express different amounts of these enzymes that metabolize ethanol to acetaldehyde or acetaldehyde to acetyl-CoA. So for those, and this is also true where different populations of people regionally across the globe will have common expression levels of these different enzymes. And so for those individuals that have a greater sensitivity to alcohol, that's largely because they will express less acetaldehyde dehydrogenase. So they're going to have a longer period of time where there's an acetaldehyde buildup in their cells. Whereas those who have a higher expression are able to metabolize it more quickly and feel the effects of alcohol as much. And so what ends up happening is that when we have an accumulation of acetaldehyde buildup, it can actually lead to mitochondrial failure, and more importantly, increase the generation of free radicals. These are these molecules that have an unpaired electron that are highly reactive that can change the function and structure of a given molecule. And so the way those free radicals are depleted are via antioxidants. There are these molecules that will react with a free radical to take on that unpaired electron and neutralize the harmfulness of that free radical species. An example of that is glutathione, but we'll talk about a few more examples of that as well. The formation of acetaldehyde can lead to adducts, these changes in the structure of a molecule that leads to the formation of what we refer to as neoantigens, where perhaps a molecule was not antigenic or promoting an immune response previously, but via changes with the free radicals in response to acetaldehyde buildup, it changes the structure of this molecule that now makes it antigenic, or that it induces an immune response. And so for an example, one example that could be perhaps collagen, whereby collagen is one of the most excessively abundant proteins in our body, but via a molecular adduct alteration, via acetaldehyde, there's the ability for now it to become antigenic where it wasn't before, inducing some sort of immune response that produces this neoantigen, neo meaning new. And so there's a change that produces a new antigen that wasn't there before. And finally, the buildup of acetaldehyde can actually trigger collagen secretion by a flavor of fibroblasts. Remember, those are the collagen or ECM producing cells in our body. There's a flavor of fibroblasts that are in the liver. Excuse me. And so that fibroblast that's resident in the liver, in response to buildup of acetaldehyde, will actually lead to excessive collagen secretion, which will contribute to scar formation or fibrosis in the liver. All of these things together that we're listing here are part of what makes up cirrhosis. And so the accumulation of acetaldehyde, the accumulation of fat, the excessive secretion of collagen, and this active immune response are all hallmarks of cirrhosis that lead to eventually liver failure. I'll take a step back and leave space for a couple of questions here. Yes.
Like when a better company increase oxidative capacity. I was just wondering, like your exercise, your diet, and you got cells that are but greater oxidative.
Just to make sure I understand, how does the combination of like a balanced diet and exercise lead to lipolysis?
Or lead to like having more mitochondria.
Having more mitochondria.
Yeah.
And so typically what ends up happening, I'll go back to that slide. So I think I've already said this before, but it's worth repeating. Our cells don't just have one mitochondria. There are many throughout our cells. And so the idea is that the ability to produce more mitochondria is a result of some, particularly in response to exercise or a certain style of exercise. There can be the ability to have more mitochondria in specific cells like skeletal muscle that are able to more efficiently burn fat, as well as the ability to produce more ATP to meet that demand, to burn the energetic substrates, in this case fat, to meet the demands of those cells, to meet demands of that tissue, and meet the demands of the entire organism. And so there's typically these things, and I think there was in your question was, how do these like broad changes at the whole person level boil, how can that lead to like this very specific subcellular change? And so what it boils down to is this need to meet energetic demands, particularly in certain tissues. And so like it goes down from like uh, these entire diet changes at the whole person level. And yeah, these whole changes, whether it's diet or exercise or both, lead to specific needs being that need to be met at the tissue level that leads to specific changes at the cellular level to meet those demands. Does that make sense?
So that feature, like a current cell will create like one item cell function at the end, or is it like a new cell get created?
I wouldn't call it a mutation. I think it might be more accurate to think of it as an adaptation, and an adaptation to meet these new demands, and the ability to function at that level, so that there's not what's a good way to say this? An ability to meet those demands, to be able to function at that level and sustain it. Does that answer the question? Yeah. Any other questions? Yeah.
Yeah, real quick. Do the physiological differences in like alcohol dehydrogenase levels in males make them more sensitive to like alcohol dehydrogenase?
Just to make sure I understand your question, does different expression levels of these enzymes make are there like sex differences in the ability to either metabolize or store fat? Is that the question?
Yeah. Wouldn't that like typically?
So what I should say is I don't know of any sex-specific differences of expression of these enzymes. I have seen like studies report that there are differences related to ethnicity. Different populations have been reported to express these enzymes at different levels. I don't know of different levels, I don't know of sex-specific differences. One thing that might be mindful to think of is that typically size is also a factor in, if two individuals who weigh, if there's a weight difference of like 75 pounds between them, consuming the same amount of alcohol is going to hit them differently. And so the idea is that also that same amount of alcohol consumption will lead to more fat storage as well. But I don't know sex-specific differences in expression of these enzymes. One thing we haven't touched on is that there's also sex-specific differences in fat storage as well. And typically, I don't think the BMI actually accounts for this either. But when we talk about subcutaneous fat and visceral fat, the amount of subcutaneous fat for an individual is going to be dependent on their sex, because typically on average, what we define as healthy is that there's a certain amount of fat that's going to be healthier for women as opposed to men. And so again, this quote unquote healthy, and what we define as healthy is going to be specific to each individual. But related to your question about the enzymes, I don't know of any sex specific differences. All right, we can press on. And again, as we're piecing all of these things together across really the last 3 lectures, if there's something that doesn't make sense that I may not have explained sufficiently, please feel free to raise your hand. Or if you're not seeing how these things fit together, again, please feel free to raise your hand. Another diet style that's worth exploring that helps piece a few things together is intermittent fasting. Intermittent fasting can be defined differently based on who you talk to or what you read. But one version of intermittent fasting is typically 5 days off, 2 days on. At least that's one version of it. And what ends up happening is that during this state of intermittent fasting, for some people, they'll just do this for 7 days a week. I don't know how they do it, but they'll do it. But what ends up happening is that it's a period of 12 to 16 hours of no food consumption, and then typically about a 6 to 8 hour window where food consumption typically happens. What's key to note about how our cellular metabolism helps explain some of the advantages of this approach is that, you know, we have glucose that we ingest via carbohydrates. There's also, again, our glycogen stores. What ends up happening during this 12 to 16-hour window is that with no food consumption, we are also depleting our glycogen stores in our liver and our skeletal muscle here. And so once those glycogen stores are depleted, this enables or triggers both fat and protein catabolism to try and produce oxaloacetate. And after that's been depleted, again, there's going to be a state of ketosis that's entered where an individual's fat tissue is broken down to produce fatty acids that leads to acetyl-CoA that's metabolized into ketones, and again, used primarily as an energy substrate in the brain and skeletal muscle. Yes. You're saying why would it switch to the liver? Is that what you said?
I feel like, if I don't eat for a while, what is that? That takes 12 hours.
Right. So it doesn't take 12 hours because there's a point where now your body is using glycogen stores.
So it uses it more.
Yes.
Oh.
So it's saying once the 12 to 16 hour time, once you reach that 12 to 16 hour time point, your glycogen stores are largely been depleted at this point. Does that help make sense? After that 16-hour time frame, then amino acids from muscle or fatty acids from fat tissue will start to be broken down to be used metabolically. Does that help answer the question? Yeah. And so, yeah, so usually after that window, there's also a state of ketosis that one enters into that, again, breaks down fat, produces acetyl-CoA that is then metabolized to produce ketones. And one of the advantages of this intermittent fasting diet is that the entering into that state of ketosis, of breaking down fats to produce ketone bodies, that actually can help make an individual more sensitive to insulin, meaning that when you will, after a period of intermittent fasting or doing this for a period of time, when you have that time frame where you're actually eating food and eating carbohydrates, your body's ability to consume glucose that then typically triggers insulin to be released, and that insulin binds to receptors on your cells to transport that glucose into the cell so that it can now be used for glycolysis. Your responsiveness to insulin goes up typically after a period of time of doing intermittent fasting. And so when an individual eats during that 6 to 8 hour time frame, it's typically going to be trying to restore glycogen stores and typically trying to replenish not just your glycogen stores, but also a lot of the metabolic intermediates that are needed to produce ATP and feed into these various metabolic pathways we've talked about. And so our individual who did a intermittent fasting diet and what you can appreciate to the right here is the range of weight loss that occurred as a response to removing vitamin water from their diet. What you're able to see is that on an intermittent fasting day where they had approximately 600 calories per day, you're able to see that over time, there was a more steady decrease in weight loss here in response to intermittent fasting. That then reached kind of a stable point where they leveled off over the course of a few months. And so, so this is an example of one diet that actually has some physiological benefits like the improved insulin sensitivity, but can actually help regulate weight loss at a more sustainable pace. Yes.
I haven't reached like minimum.
Yeah.
Like between, I guess what like your whatever the 600 calories like storage, and that would be like equal.
You're good.
Well, to like body's like energy.
Yes, exactly. And so you can imagine if this was an individual who let's say they did it, so they did this out to about 110 days. If at day 110 they continue doing this and change their activity level to be a lot more demanding, you'd be able to see, and if they change nothing about their diet, you'd see that while they reach equilibrium here, then they'd probably start losing more weight because again, um, they've kind of determined here what their steady state is, for lack of a better phrase, with that intermittent fasting. That would then change if you have a higher demand of a higher energetic demand. And so this kind of touches back on a question earlier, but a lot of this kind of boils down to the meeting of demands. What is what is energetically needed? What are the substrates available? And how can we leverage those substrates that are available to meet those specific demands? So one thing that's worth touching on is that I think some of you have included in your projects to some degree or another is this idea of oxidative stress. And so oxidative stress it's both a normal part of biology as well as a critical part of some diseases. But it's this idea of an imbalance between oxidizing compounds and our body's ability to produce molecules that help neutralize some of these oxidizing molecules to avoid or limit damage. And so during oxidative phosphorylation, where we have our reducing equivalents that undergo oxidation and lose their electrons, that's then passed across complex 1 to complex 3 to complex 4. It's not a perfect system where these electrons are perfectly passed over. There is sometimes the leakage of those electrons out of these complex proteins. Their final destination is to get to an oxygen molecule to produce water, to help produce water. But sometimes what can end up happening is that there is an early leakage of those electrons that too quickly get to an oxygen molecule that leads to the formation of these molecules that we refer to as reactive oxygen species. The two most commonly discussed reactive oxygen species are superoxide and peroxide. And in addition to that, there's not just reactive oxygen species, but there's also reactive nitrogen species. Both nitric oxide, which we've touched on a little bit in previous lectures, but also peroxynitride as well. And so the list is more, there's a longer list than these four, but these are the four that are most commonly referred to in cell biology. And again, these are oxidizing compounds with an unpaired electron that are highly reactive and that in some doses are just a normal part of biology. It happens. Nothing about biology is perfect. So the notion that there is some electron leakage is going to happen. But when there is excessive amounts of these oxidizing compounds that we refer to with these unpaired electrons that we refer to as free radicals, they can be highly reactive and alter other molecules that can lead to damage. And so during aerobic metabolism, where we're going to the TCA cycle and then oxidative phosphorylation, there is a constant leakage of electrons to some degree. So there's going to be the production of these reactive oxygen species. Again, it's a normal part of biology, and we have mechanisms to limit their effect. And we'll talk about those mechanisms in a second. But again, what I want to try and highlight here is that we have these reactive oxygen species and nitrogen species that are free radicals that have these unpaired electrons that are highly reactive. And they are in small doses, in a limited amount, a normal part of our biology. And excessive amounts can lead to damage and are likely a part of the etiology for some of the diseases you guys are studying in your projects, as well as some of the diseases we've talked about in class already. And so as I described again, the passing of these electrons that come from our reducing equivalents, NADH and FADH2, it's not a perfect system as we're going from complex 1 to complex 3 to complex 4, where again, its final destination is to produce a water molecule with this oxygen here, but there's going to likely be leakage out at complex 1 or out at complex 3 that produces the free radicals and those oxidizing compounds or those oxygen species that we were just talking about on the previous slide. And so as an example here, we can have a electron leak from the reduced FMN molecule that we see in complex 1. And again, a similar electron leakage is also happening to some degree at complex 3 as well. What happens or what the consequences are when we talk about the damage that are associated with these free radicals or with these reactive oxygen species is that they can alter other molecules. So as an example, we have superoxide here with arginine that ends up carbonylating this arginine. It can carbonylate a protein whereby it replaces that amine with a carboxyl group there. And for amino acids or for proteins, it can actually change their, we are changing their chemical structure. We are changing their physical structure, and finally will lead to changing their function as well. And so what ends up happening is that this can actually be more deleterious when this also happens with lipids. Lipids can undergo a carbonylation modification via these reactive oxygen species that alters their structure and alters their function. And so, again, we're going from, I guess, we're increasing in degrees of severity here because in addition to lipids and proteins, it can also damage our DNA as well. And so there's going to be the ability for these reactive oxygen species to promote base pair damage or base pair changes that can have really harmful or really deleterious effects that are likely, again, probably part of the etiology of some of the diseases that we've talked about, as well as the ones that you guys are exploring in your projects. Given how many different molecules can be altered by reactive oxygen species or free radicals, there needs to be a way to mitigate their effects and a way to eliminate these free radicals. And the way in which that happens is by antioxidants. Antioxidants are functionally almost like a scavenger enzyme whereby they will have a substrate on them that's ready to take up these unpaired electrons and neutralize that free radical and neutralize that reactive oxygen species and render it harmless, for lack of a better phrase. And so one example that we briefly touched on a few slides ago is glutathione. And so glutathione is one example of an antioxidant whereby we have an unpaired electron that's added to an oxygen molecule to produce superoxide. It can produce a hydrogen peroxide, and that's the way we want it to go. And so we will typically have some sort of antioxidant, in this case, superoxide dismutase, which will take that superoxide and alter it to produce hydrogen peroxide, and via the activity of another enzyme, produce water and oxygen. What can also happen is via this glutathione peroxidase, take that hydrogen peroxide and produce water as well as glutathione intermediate. And so the take home here is that largely these antioxidants will help work to try and neutralize the free radicals on these reactive oxygen species to render to change them so that they are no longer able to induce damage to proteins, lipids, or DNA. Oxidative stress can be a bit of a bear to study. Taking a moment to step back and talk as a researcher now, it can be really hard to investigate as a scientist, largely because, for example, if we just focus on one type of reactive oxygen species, it can alter a few different kinds of molecules. It can become a variety of different molecules, and what it can be mitigated by is going to be not just superoxide dismutase, but can also be a variety of different antioxidants as well. And the degree of complexity expands greatly when we consider what can happen based on the different molecules that this reactive oxygen species can become. And so not only is there a complexity when going from what it can affect to what it can become and what it's mitigated by, but there's also a lot of contextual factors that dictate these responses or dictate these processes as well. That can be the presence of ions, the changes in pH, as well as other compounds as well. I'm going to step back and give space for questions here. Yeah.
What do you say?
It is the final destination of that, of those electrons that are donated by the reducing equivalents. And so it helps, they would say this. It helps produce a molecule that's, rather than producing some sort of like harmful reactive oxygen species, it can bind to oxygen and produce water instead. That's just shuttled out and shuttled out of the mitochondria. But it is largely just a non-harmful product as opposed to producing a reactive oxygen species. Does that make sense?
That's the point, that's water, it's more that's not, like our.
Yes.
Why are, reactive oxygen the one years,
It's only formed during aerobic respiration because that's when that electron transport chain is active. During anaerobic, we don't have oxidative phosphorylation. So you don't have that risk of electron leak during anaerobic respiration. All right. So if our basically lecture series on metabolism really caught your interest, we have a few folks in our research community who are actually leaders in the field. Thurl Harris, particularly, is focused on insulin signaling and fat synthesis, as well as how it relates to cardiovascular medicine. Zhen Yan is a really key leader in the field, looking at mitochondrial function as it relates to exercise and skeletal muscle plasticity. A couple of folks are outside the UVA community, but leaders in the field. Alyssa Hastie's exploring immunometabolism, particularly that weight cycling idea that I discussed, and how that relates to how that affects your immune system, how it functions. And Leah Bellis is a BME professor at Temple up in Philly, who's actually engineering unique biomaterial models to study adipose tissue, adipocyte biology, and differences in sex-specific differences, as well as the variety of adipose cells as well that we have. So that concludes today's lecture. We have that survey that I talked about a second ago. So I'm gonna actually give you guys time to take that survey. Shannon Barker said it's like a 20-minute survey. I suspect that's probably longer than what it actually is. But the idea, again, is that it's meant to be a survey that all BME students take at the end of the year, so that we can understand what needs need to be met in the BME curriculum. Did you have a question a second ago?
I was gonna ask.
It is anonymous. Thank you. So it's anonymous, and there's also demographic data that you can put in. You are not obligated to provide that data, but that option is available to you. Yes. If you are a first year, you do not have to do this. This is primarily for BME majors. So I guess if you're a first year, you're dismissed. I'll see you on Thursday. If you're not a first year, please take the survey. It really is helpful if you take the survey. There's a lot of, I can probably stop recording here, but there's a lot of work going on behind the scenes right now to help try and improve the BME curriculum and totally revamp it to make sure that whatever changes need to be made to ensure the success of all of our students is able to happen. And so we've had three teaching retreats already based on previous year's survey. And so it's super helpful for our second, third year, and fourth year students to provide feedback on the survey. First years, you can go. Yep.